Good morning, everyone. Let us talk one more topic under biochemistry, that is regulation of blood glucose level. So, what are the objectives of our presentation? We have to know regarding what kind of regulation is going to occurring in our body for controlling our blood glucose level. So, regulation of blood glucose level, under these things we have to talk about maintenance of blood glucose in fed state, that is hyperglycemic condition, maintenance of blood glucose in fasting state, that is hypoglycemic condition. So let us come to the what do you mean by regulation of blood glucose level? The normal blood glucose level is 70 to 100 milligram per deciliter. Levels above the normal range are termed as hyperglycemia. Those below are called hypoglycemia. So we have to understand here two kind of terminology. One is hyperglycemia, another one is hypoglycemia. The normal blood glucose level is 70 to 100 milligram per deciliter. When these levels are raises, that is known as hyperglycemia. When these levels are below, that is known as hypoglycemia. After the ingestion of carbohydrate meal, it may be raised to 120 to 140 milligram per deciliter. After eating, once we had the food, our blood glucose level used to be raising. So it can be acceptable until 120 to 140 milligram per deciliter. And the liver is the organ primarily responsible for controlling the concentration of blood glucose. So which organ is responsible? The organ is liver. Liver is the organ primarily responsible for controlling the concentration of blood glucose. Liver can rapidly take up and release glucose in response to altered level of blood glucose. Whenever our body is undergoing altered level of blood glucose, the liver is helpful for maintenance of the normal blood glucose levels. The uptake and release of glucose by liver is regulated by some of the hormones. How the liver is controlling this? With the help of some of the hormones. The two major hormones controlling blood glucose levels. What are they? One is insulin, other one is glucagon. Insulin is known as hypoglycemic hormone, whereas glucagon is known as hyperglycemic hormone. Then the first condition we have to talk about maintenance of blood glucose in fed state, that is known as hyperglycemic condition. Normally there is an increased blood glucose level shortly after each meal. That is also known as postprandial hyperglycemia. So what they are telling here, there is an increased blood glucose level. There is a increasing of blood glucose level shortly after a meal. Once we have a meal, our blood glucose level will be raising. That is known as postprandial hyperglycemia. Increased levels of circulating glucose releases insulin by beta cells of islets of the Langerhans. What is happening? There is an increased level of circulating glucose. Once there is a glucose is increasing, which releases the insulin by beta cells of islets of the Langerhans. This hormone decreases the blood glucose level in a different methods. By stimulating the active transport of glucose across cell membrane. So how it is going to be controlling? Stimulating the active transport of glucose across cell membrane of muscle and adipose tissue, but not the liver. Glucose is rapidly taken up to liver as it is freely permeable to glucose through glucose transporter. So how this? The glucose is rapidly taken up into the liver. So whenever blood glucose level is increasing, the remaining excess blood glucose level will be entering into the liver in the manner or with the help of the permeable to the glucose or GLUT transporter, that is known as glucose transporter. In the liver, insulin increases the use of glucose by glycolysis by inducing the synthesis of key glycolytic enzymes. So in the liver, what is happening? Insulin increases the use of glucose. Insulin is going to be increases for the use of the glucose by glycolysis by inducing the synthesis of key glycolytic enzymes. What are they? Glucokinase, phosphofructokinase, and pyruvate kinase. So what is the meaning of your glycolysis? The breakdown of glucose by enzymes releasing energy and pyruvic acid. Then this glucokinase is important in regulating blood glucose after meal. So once the meal is completed, this glucokinase is important in regulating our blood glucose. And like the hexokinase, hexo means of extrahepatic tissue. Glucokinase of the liver is not inhibited by glucose 6-phosphate. Then glucokinase increases in activity whenever blood glucose concentration is higher than the normal levels and seems to be specifically concerned with the glucose uptake into the liver after a carbohydrate meal. So here what is happening? This glucokinase increases in activity. This glucokinase is going to be increasing in activity whenever blood glucose concentration is higher. When there is a blood glucose concentration higher, this glucokinase is going to be initiated. The normal levels and seems to be specifically concerned with glucose uptake into liver after a carbohydrate meal. Sorry. In the muscle and the liver, insulin stimulates glycogenesis by stimulating glycogen synthase and thereby leading to suppression of glycogenolysis. In the muscle, what is happening? In the muscle and the liver, insulin stimulates glycogenesis by stimulating glycogen synthase and thereby leading to suppression of glycogenolysis. So lysis means breakdown. So uh, glycogenesis, the formation of glycogen from sugar. Insulin inhibits gluconeogenesis. So what is the meaning of this gluconeogenesis? Glucose, neo means new, genesis means production. The insulin is going to be controlling for availability of the glucose, and glucose will not be available in the blood by suppressing the action of key enzymes of gluconeogenesis. For example, pyruvate carboxylase, PEP carboxylase, that is also known as phosphoenvelope pyruvate carboxylase, and fructose 1,6 biphosphatase and glucose 6 phosphatase. In adipose tissue, glucose is converted to the glycerol 3-phosphate needed for the formation of triacylglycerol, that is lipogenesis, and inhibits the lipolysis by inhibiting hormone sensitive lipase. Insulin increases protein synthesis and decreases protein metabolism, thereby decrease the release of amino acids. So these all mechanisms are responsible for a drop in the blood glucose level and create hypoglycemia. So what is the meaning of this protein catabolism? It is a breakdown of proteins into smaller peptides and ultimately to the amino acids. Then another condition is maintenance of blood glucose in fasting state, that is known as hypoglycemic condition. Within about 1 hour after a meal, blood glucose levels begin to fall. By 2 hours, the level returns to the fasting range. Fasting range is 70 to 100 milligram per deciliter and leads to hypoglycemia. What's the concept of here? After feeding or after we had a lunch, our blood glucose level going after 2 hours, after eating, within 2 hours, our blood glucose level has to become normal ranges. That is at least 70 to 100 milligram per deciliter. This leads to the hypoglycemia. For the brain, the erythrocytes, the bone marrow, the renal medulla, the peripheral nerves, glucose is major fuel. So these all the organs are utilized by the glucose is the major fuel which has to be supplied for their energy needs. Therefore, it is essential to maintain the blood glucose level within normal ranges. The blood glucose level is maintained within the normal range by the following ways. How it is managing its normal ranges? The decrease in blood glucose, that is hypoglycemia, causes decrease in insulin secretion and stimulate the release of hyperglycemic hormone. For example, what are the hyperglycemic hormones? Glucagon, epinephrine or adrenaline, glucocorticoids, growth hormone, and adrenocorticotropic hormone and thyroxine. Glucagon. So how it is going to be helpful? Glucagon opposes the action of the insulin. Glucagon is secreted by alpha cells of the pancreas in response to low blood sugar level in fasting state. Glucagon causes an increase in the blood glucose concentration in several methods. It acts primarily in the liver. As in the liver, it stimulates the glycogen breakdown and inhibits glycogen synthesis. The glucose formed by the breakdown of glycogen is then released from the liver into the blood. So what is happening here? The glucose formed by the breakdown of glycogen is then released from the liver into blood. The liver glycogen is capable of maintaining the blood glucose concentration at the normal value for 8 to 12 hours. The liver glycogen is capable of maintaining the blood glucose concentration at normal value for 8 to 12 hours. Then glucagon enhances gluconeogenesis. Glucagon enhances gluconeogenesis. Newly or excess releasing of the glucose production is going to be identifying from amino acids and lactate by inducing the action of key enzymes of gluconeogenesis. So lactate formed by oxidation of glucose in skeletal muscle is transported to the liver by lactic acid cycle. Then other hormone is epinephrine and adrenaline. It's secreted by adrenal medulla. It stimulates uh, glycogenolysis in the liver and muscle. In the liver, glucose is the main product leading to increase in blood glucose. Then other hormone is glucocorticoids. These hormones are secreted by adrenal cortex which stimulates the gluconeogenesis by increasing the activity of enzymes of gluconeogenesis. Then another one is anterior pituitary hormone. So growth hormone antagonists the action of insulin by decreasing the glucose uptake in the muscle, and STH decrease glucose utilization by the tissues. Then thyroxine. How the thyroxine is placing a role here? Thyroxine accelerates hepatic glycogenolysis with the consequent rise in blood glucose. Then renal control mechanism. Renal means kidney. When blood glucose rises in a relatively high level, the kidney also exerts a regulatory effect. Glucose is continuously filtered by the glomeruli. So what is happening here? So glucose is continuously filtered by the glomeruli, but it is normally reabsorbed completely in the renal tubule. The capacity of the tubular system to reabsorb glucose is limited. If the blood glucose level is raised above 180 milligram per 100 ml, complete tubular reabsorption of glucose does not occur, and the extra amount appear in the urine causing glycosuria. So what is the concept of here? If blood glucose level is raised above 180 milligram per 100 ml, complete tubular reabsorption of glucose does not occur. The tubules will not going to be in the nephron. The tubules will not going to reabsorb the glucose because there is excess availability of the glucose in the blood, and that does not occur, and extra amount appears in the urine. The excess glucose whatever is available, that is going to be eliminated by the kidneys causing glycosuria. In the previous classes we have discussed regarding what is glycosuria. Glycosuria means presence of glucose in the urine. Then that's by excreting extra amount of sugar in the urine during hyperglycemic state and reabsorbing sugar during hypoglycemic state. The kidney helps in regulating the level of glucose in blood. So let us summarize our topic. So far we have discussed regarding how our body is going to be regulating our blood glucose level. So normal blood glucose level 70 to 100 milligram per deciliter. In some conditions the blood glucose level may be increases, and some condition the blood glucose level may be decreases. So the major two hormones are insulin and glucagon. Insulin will be reducing our blood glucose level, whereas glucagon will be increasing our blood glucose level. and some of the hormones is also is placing a role. Here majorly we having two conditions we have discussed. The first one is maintenance of blood glucose level in fat condition, that is known as hyperglycemic condition, and other condition is maintenance of blood glucose level in fasting state, it is known as hypoglycemic condition. I hope my dear students you understand the concept of regulation of blood glucose level. Have a nice day. Once again we'll meet.